In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you, dear students? I hope you all are doing well and you are safe and sound with your health at your home and enjoying your studies with virtual learning system of Pakistan International School Types. Dear students, I welcome you in Pakistan International School Types virtual learning classes, and this is a class of biology for grade second year. Dear students, in this lecture, we are going to continue chapter number 21, that is about development and aging. And before we have seen in that the first stages of development, that is cleavage, gastrulation and neurulation formation. Now we will see how the development is controlled in this lecture. Let's get started. Dear students, we see control of development, like control of development. um, that question came in the minds of scientists, that how does a developing embryo generate the multitude of many cell types of a completely multicellular organism from the starting point of a single diploid nucleus of a zygote. So that is a complicated question, that how it can be happen that from a single single diploid cell, then multicellular organisms can be formed. How it can be happen? So there are we can see some of the views, and we can see the very first hypothesis which came after this thinking and some observations, that Rocks Wiseman, two scientists, they gave the hypothesis that genome gradually broken into smaller and smaller units, like the DNA of that cell just got broken into smaller and smaller units. Finally, only the information required to impart the characteristics of a single cell type remain. So if the different types of cells are going to be formed, so that genetic information just going to be decrease and decrease in that cell. And like for example, there is a respiratory system is going to be formed, so the information and the genetic material about formation of respiratory system is just going to be get forward into the cells. So early embryologists saw this as an explanation of differentiation. Like differentiation of cells is done. Differentiation of cells means different cells make different organs and organ systems. So hereditary material was parceled out to the cells. Specific hereditary material. So whether it is a logical, whether it is experimentally proved or not, some other scientists have discovered, or they have done some other experiments regarding differentiation of the cells. So how this multicellular organism is formed. So in that, we will see two controlling system. Number one is role of nucleus and cytoplasm in the controlling of development, and the other we will see embryonic induction. So first we'll talk about that nucleus and cytoplasm are having too much great role in differentiation of the cells or starting the development. So Hans Driesch, uh, he just performed several experiments, and in 1892 he proposed that based on experiments on sea urchin, after first cell division, what he did, he shook the cells apart. Shook means, with a shaking movement, he just make the cells apart. That was on two cell stage, like first cleavage, after first cleavage, and then place them separately, you can see. And he did experiment on sea urchin. So instead of producing partial embryos, the cells produced normal embryos that were half size. Like size will be just a smaller half of that, but the normal organisms are being formed from these two separated cells, which are separated from two cell stage at time of first cleavage. So like Drish concluded that both cells contain all the genetic information which is present in the nucleus to form the new original zygote. But this experiment did not settle the argument, because many embryologists yet they believed that even if all the cells contain the complete genomes, so the nuclei might become progressively modified in some way to dispense with the information that do not use in forming differentiated cells. So like the specific information is utilized for specific functioning or differentiation of the specific type of cells. The scientists yet they just believe on that all hypothesis. Hans Spiemens, that is another scientist. Hans Spiemens experiment, it is very important experiment as well, in which he totally disproved the Rogge-Wiseman hypothesis. What he did in his experiment, the first step he has done, that salamanders, zygotes, salamander is like amphibian, and the long tail like lizard like, but it's amphibian, were about to divide. So he tied the cell of that salamander, that is zygote, and when it is going to be divided, so he divided one cell, that like zygote, not before cleavage, with a human ligature, because human ligature is very thin and sharp. So just he tied them with a human ligature, but did not completely separated the zygote into halves. So what happened? Nucleus was on one side and the other side only contained a cytoplasm. Nucleated side divided many times at about 16 cell stage, and nucleus wandered across the anucleate side. So only then did the cytoplasmic side divide. So both sides developed normally, and it is demonstrated that no nuclear material had been lost after many nuclear division. So here is its practical view, that autonomous specification, like how that is done naturally and how it is done by by inducing. So he demonstrated nuclear equivalence or nuclear induction in that. So it is done on the newt or salamander cells. So like you can see in this diagram, he divided at the 8 cell stage, and with the human ligature. So here is this is only cytoplasm, no nucleus is there, and here nucleus is there, so it starts to divide divide as normally. But at 16 cell stage, what happened? One of the nucleus wandered and moved to the other side, because that was loosely tied with ligature, human ligature. So the nucleus migrates to other side. Here the cell, normally it started to divide and it becomes the normal organism, and on the other side nucleus has been moved later on, but the time is delayed, but the due to the movement of the nucleus, that is done properly, and the organism is delayed formation, but that is completely developed here. The same experiment is repeated, but with a different, we can say, uh, just a slight difference. Here you can see in this one, this white area. This white area, this is called gray crescent. In the first experiment, gray crescent, when he tried with ligature, that is horizontally, so gray crescent is divided equally on both of the sides. So both of the organisms are normal. But on the second like experiment, when he just like tied it, here it is tied horizontally, here it is tied vertically. So horizontally, gray crescent just comes on one side and not the other side. This nucleus is going to develop, and it at 16 cell stage, then one nucleus is moved towards the other side where the gray crescent is present. So here, by the arrival of nucleus, this cytoplasm or this cell starts to divide, and then this division is normal, and a normal organism is developed. But on the other side, where no gray crescent was found, so there was this, we can say, uh, just the belly portion was there. Belly portion means there's just the meat over there, no organs and nothing, no, like incomplete, this no organ development is there. And on the other side, where the gray crescent and the nucleus was present, so complete development of the organism is done. It means that gray crescent is also important. What is gray crescent? Gray crescent means it is having the structure like this and a slightly grayish in color, but that is without pigmentation. If we see the structure of egg, this is animal pole and this is a vegetal pole. A vegetal pole means here most yolk is present, is a most and like less yolk is present or no yolk is present, so it divides very rapidly, and vegetal pole divides slowly. This crescent is present like dark surface is there, but that is without pigmentation, without any type of animal, and some molecules are not presented. But there is a specific and very important area of the cytoplasm, which here you can see in this diagram, which helps in the formation, or that is, we can say, the inducer, or as an organizer, which organizes or induce the development of the organism, and it gives signals to other cells to develop into a proper individual. If gray crescent is there, so organism can develop properly. If gray crescent like is divided like this, gray crescent and nucleus on both of the sides, so both organisms like receives gray crescent and they develop normally. But if you see here, gray crescent on one side, so organism develop normally, and other side where gray crescent is not present, so that will be just belly or a mass of the cells. So not only, okay, after that he decided, and it is concluded that not only nucleus, but that gray crescent is also very important for the induction process or for the development of the individual. So all cells contain the same nuclear information. So this is first conclusion of that experiment that disproving the Rox-Wiseman hypothesis. The second one is that cytoplasm in the area of gray crescent must contain information essential for normal development of the organism. So there are two major ways by which cells become committed to particular fates, like which type of cell is going to develop into which type of organ system. So here the cytoplasmic segregation of determinative molecules during cleavage, that is very important. Like cytoplasm, when it divides, it must have those determinants which is important like when the cell is dividing, so that we can say helps to differentiate the cells into different categories. So they are also called morphogenetic determinants, that these are the proteins, some sort of molecules which are very important, and these are also called determinative molecules or morphogenetic determinants, which determines the morphology, or we can say the structure of the required organ system. Number two, interaction with the neighboring cells. That is also very, very important. That is also called inductive interaction. Like the cells signals or interact with one another, with the neighboring cells, and induce the interaction between one another and induce the process of development. So here it is proved that nucleus and cytoplasm both together controls the developmental process. Now we will see embryonic induction. That is also very, very important, like how the organism or which part of the cells are going to induce the process of embryo formation. So induction is the process by which one group of cells produces a signal that determines the fate of the second group of cells. So first group of cells just determine the fate of the other group of cells. That is called induction. So this implies both the capacity to produce a signal by the inducing cells and the competence of the responding cells to receive and interpret the signal via a signal transduction pathway. So that is, we can say, along a process like they are like the cells of the first cleavage cells are inducing like the responses on the other cells to produce a specific or we can say the differentiated cells in an organism. So most of the experiments are and studies are done in the amphibians and like which are vertebrates. There are two major inductive events during early toad development, like that is a category of frog and amphibian. So that is mesoderm induction and neural induction. So how this process is done, that is done. So the great work of experimental embryology is done by Spearman. So Spearman and his colleague or his student Mangold, they have done the experiment and performed many types of transplants at the early gastrula and the late gastrula stages in nude embryo. So these experiments showed that the most cases the cells of the embryo are not committed until at least the last gastrula stage. So after the gastrula stage or in the gastrula stage, that is determined that which type of cells are going to induce which type of like the response in the other cells. Here's Hans Spearman and Mangold in 1924. They are awarded by Nobel Prize on this performance in 1935. So what they did, they took a piece of lip of blastopore lip of blastopore of the gastrula stage of the amphibian embryo and grafted it into the wall of the flank of another gastrula at a slight distance from the host blastopore, thereby induced a secondary axis of the polarity in the host that eventually develop into the secondary embryo. So you can see this is the late gastrula stage. Late gastrula stage means the three germinal layers are formed here, like ectoderm, mesoderm, and endoderm. So blastopore, this side actually is the area from which the cells at the start of gastrula, early gastrula cells are going to move inward. So that this area just transformed into blastopore. Blastopore, like the hole from which all the blastomeres just move inward to form the three germinal layers. So, but here that is called dorsal lip of blastopore. Dorsal lip means this is dorsal side and this is ventral side. So dorsal lip of blastopore here. That is taken from one embryo or zygote after like in the late gastrula stage. So there is primary axis in which this blastopore has already been developed, and in that we can see the neural tube is developed, notochord is there, and somites are there, mesoderm is there, and just rudiment of gut is going to form. Like this is called, we can say the cranial end. So what they did, they took the dorsal lip of blastopore from one embryo and just grafted on the other side or the back side of this embryo. At the back side of embryo, when he grafted this one, so what he saw that the same process is going to be done, and again it induced the secondary axis of that embryo. So here also the neural tube and the notochord and the rudimentary gut is going to be formed. So now we will see that Spearman's experiment, in that the dorsal lip is said to be as organizer of Spearman and Marigold. So he gave the conclusion that dorsal lip acts as primary organizer. So based on their famous experiment, Hans Spearman and Mangold concluded that blastopore's dorsal lip is an organizer of the embryo. That is called primary organizer. So the Spearman organizer initiates the induction that results in the formation of notochord, neural tube, and other organs. So at the end, just he observed that from both of the dorsal lip, two same organisms are going to be formed, but they are attached with one another because the dorsal lip of blastopore that organizes or induces the formation of nervous system, notochord, neural tube, and like all the neurons. So here you can see again the development of that. So that is called primary induction, or that is done by primary organizer. So that is the first event in the development. Subsequent studies showed that many other cell types originate by inductive interaction, a process called secondary induction. For example, cells of the neural plate uh, induced neural crest into the embryo, as we have seen in neurology. Neurulation process, that when this neural tube is formed, and this is neural tube, so this neural plate just induced the formation of neural crest cells. These are neural crest cells, and these neural crest cells are going to be formed some other we can say this some other some other like cranial, spinal, and sympathetic ganglia and associated nerves also. And that is also like forming peripheral nerves, medulla of the adrenal gland, teeth, skull, bones, and so many other different cell types that some have proposed considering neural crest as the fourth germ layer, because that induced the formation of lots of other, we can say, the organs or tissues. So that is proved now that embryonic induction is done by the dorsal lip of blastopore, and we have also seen that nucleus and cytoplasm in the Greek crescent is the part of the cytoplasm. So they are very, very important for the development of an organism. Dear students, that's it for today. I hope you enjoyed and understood the lecture properly. We will continue, inshallah, with the this chapter with the next topic, human embryonic development. Till then, Allah Hafiz, wish you all the best.